To study whether defects in long-distance axonal transport are present in neurological diseases such as Alzheimer's disease or Huntington's disease, axonal transport of transport vesicles is imaged in live Drosophila larvae. Here, wandering third instar larvae that express a GFP-tagged vesicle protein are dissected on a gel cube, then transferred to the microscope for in vivo imaging. Fluorescence microscopy is then used to track vesicle transport within the axon.
The main advantage of using this technique over existing methods is to visualize live, undisrupted movement of synaptic vesicles and larval axons.
Prepare for the dissection by ensuring that the following supplies and reagents are at hand: dissection buffer, a Sylgard Cube cut to 1 inch wide, 1 inch long, and 1 centimeter high, placed on a glass slide. Extra short dissection pins. The sharp bottom portion minucium pins works best in dissection. The correct length can be attained by inserting longer pins into the gel and cutting off any portion of the pin that protrudes. You will also need 1 pair of sharp microdissection spring scissors and 2 sets of fine-tipped forceps. Use a small metal spatula to transfer wandering third instar larvae expressing a GFP-tagged vesicle protein.